Welcome back, students. Welcome to Biochemistry class. We are discussing protein chemistry and we have already covered classification of amino acids and ampholytes and isoelectric point. Now in this third and last part of protein chemistry, we will discuss about structural organization of proteins. So the learning objectives will be, we will discuss primary, secondary, tertiary and quaternary structure of protein. What are they and how are they different and what is their importance. Then we will discuss about denaturation, protein folding, role of chaperones and disorders related to protein folding. And we will also discuss electrophoretic pattern for various disorders. So first of all, how are proteins made up of? So proteins are made up of amino acids linked together by peptide bond. So is that all for protein structure, that amino acids are arranged in linear structure? No. For proteins to be functional, first they need the primary structure in which there is a linear sequence of amino acid joined together with peptide bonds. Then there is a secondary structure and further it is folded into tertiary and quaternary structure to be active. So let's discuss these structures one by one. So first is primary structure. So we know that if by the process of translation, amino acids are arranged in the sequence as dictated by DNA and then RNA. So this linear sequence of DNA of amino acids joined by peptide bonds forms a primary structure. So this is a primary structure. So this primary structure describes a number of amino acids, type of amino acids and sequence of amino acid, and they are specific for each protein. And so the bond in primary is covalent peptide bonds between amino acids which together form a primary structure. So this primary structure, this arrangement of amino acid is determined by the genetic information of the DNA. So if DNA base sequence will dictate mRNA and mRNA will dictate which amino acid to be joined in with what sequence. So it is all dependent on the DNA. It is dictated by DNA and its arrangement of amino acid will form a specific protein. The specific information that determines the finally 3D form, that is final structure of the protein, is also due to primary structure. So it will dictate the folding also, which determines the function of protein. And even if there is slight change in primary structure, that is modification of even single amino acid residue, can lead to loss of its biological activity. So for protein to be functional, the proper sequence of amino acids is required, and even if one amino acid is deranged or different, it can lead to loss of biological activity of protein. So it's very important. Then denaturation, this is important. Denaturation does not affect the primary structure. So even there is high temperature or alkali condition or chemical conditions, then also the primary structure will remain intact. And this is important as it is frequently asked, which structure remain intact on denaturation, that is primary structure. So there is no, it is not, denaturation is not accompanied by hydrolysis of peptide bonds. So they remain as such. So then coming to secondary structure. Now the primary structure is only linear linking of the amino acid. But do they exist as such? No. They are further folded into either alpha helix or beta sheets. So depending upon their folding, they either form helical structure like this, or they can be folded as we fold clothes like that form. They will form beta sheets. So linear sequence of amino acid does not extend as such. It may coil, that is alpha helix. For example, alpha keratin of here, or it may either form a helix or fold into beta pleated sheets like beta keratin in feather of birds. So the which bonds are required here? So here there is mainly hydrogen bonds. So mainly hydrogen bonds helps in either coiling of the alpha helix of the primary structure into secondary structure or folding into beta plated sheets. So how it is possible? Because there is formation of inter and intra-chain hydrogen bonds, which helps in coiling or folding of the primary structure of protein to secondary structure. So along with hydrogen bonds, there can also be disulfide bonds for some amino acids which contain sulfur. So the secondary structure mainly consists of hydrogen bonds which helps them into folding of into alpha helix or beta pleated shape structure. Now there are certain amino acids which you should know which disrupts the alpha helix. Examples are proline. Now proline, since the peptide bond nitrogen or proline lacks hydrogen atom. So as we have discussed, it is NH group, either the NH2 amino acid. So it is incapable of forming hydrogen bonds. So it can be accommodated either in the first turn of the alpha helix, or it will disrupt the structure and form a bend there. So proline disrupts the alpha helix structure because it cannot form hydrogen bond. So if it is present in the first coil, it is not needed to form hydrogen bond with the above coil, then it can work. Otherwise, it will produce a bend. Similarly, glycine, which is the smallest amino acid, it also induces bends within alpha helix. So these are the examples of some amino acid which it disrupts alpha helix, and you should know. Then this was about secondary structure. So secondary structure, we have discussed that the primary structure of protein is folded either into helix or into beta plated sheets. And next is tertiary structure. But in between primary, secondary and tertiary structure, there is one structure which is known as supersecondary protein structure, also known as motif or folds. So they are combination of alpha helix and beta sheets which are connected through bends or loops to form intermediate patterns like turns, bends, loops, and they are known as motifs or folds. So turn and bends are short segment of amino acid that join two units of secondary structure. That is, two units of secondary structure, either beta turns or alpha helix, are joined by a short segment of amino acid. Similarly, for loops, long segment of amino acid that join to secondary structure, they are known as loops. So turn and bends have small short segment of amino acid which joins two secondary structure, whereas loop is a long segment of amino acid which again joins two secondary structures. Such are known as supersecondary structures and motifs. Motifs have already been covered in genetics also. So these are the diagrams. So you can see helix, loop, helix, beta unit, hairpin like structure, Greek key structure, alpha beta barrel structure, beta meander structure. So these are the examples of supersecondary structures. So they are in between secondary and tertiary structures, and they are also known as motifs and folds. Then coming to tertiary structure. Now we have discussed that there is a primary structure which is a linear arrangement of amino acids which is then folded into either helix or beta plated sheets. Then further also they are again folded on on themselves. The big coil folds upon itself to form a hollow woolen ball-like structure. So it again is folded in such a way that it forms a woolen ball-like structure, and here hydrophilic amino acids are positioned exterior, whereas hydrophobic are positioned interior of that ball. So this is a tertiary structure, and this structure is essential for biological activity. So tertiary structure is required for biological activity of the protein, and it is formed by folding on of the coil on itself, such a way that in exterior of this woolen ball which is formed by folding of alpha helix, there are hydrophilic amino acid, and the interior there are hydrophobic amino acids. Because hydrophilic can easily form hydrogen bond and ionic bond and can interact with other proteins. So example is myoglobin. So this tertiary structure is formed by folding of the secondary structure into woolen ball-like structure. And it is very important that it is required for functioning of the protein. So which bonds are involved in tertiary structure? So we have discussed in primary structure, there are mainly peptide bonds, that is form bonds between amino acids, whereas in secondary structure, there are mainly hydrogen bonds and disulfide bonds. Now in the tertiary structure, there are many bonds like hydrogen, disulfide, hydrophobic bonds, ionic bonds, and van der Waals forces. So there are many types of bonds which act in tertiary structure. So then after tertiary structure is a quaternary structure. Now what is the difference between tertiary and quaternary structure? Now tertiary structure forms one unit, but many such units when joined together to form a protein, then it is known as quaternary structure. So different subunits assemble together and forms a functional structure. So like hemoglobin, part of hemoglobin consists of 2 alpha and 2 beta subunits. So it consists of 2 alpha and 2 beta subunits of hemoglobin. So hemoglobin is formed by 4 subunits. Similarly, lactate dehydrogenase enzyme is formed of 4 subunits. So this protein can be active only if these subunits come together to form a final protein, that is active form of protein, that is quaternary structure. And the bonds here are same as that of tertiary structure, like hydrogen bond, disulfide bond, covalent bonds, hydrophobic ionic bonds. So all these van der Waals forces, so all these bonds are same as in primary and secondary structure. So if we discuss, the primary structure is linear arrangement of amino acid and the bonds are peptide bonds. Secondary structure is coiling of the primary structure in either into alpha helix or beta plated sheets, and the bonds are hydrogen bonding, mainly hydrogen bonding, inter and intramolecular hydrogen bonding along with disulfide bonds. Then there are supersecondary structures which are intermediate between secondary and tertiary structure, of which joins the two secondary structures, loops or folds. Then there is tertiary structure, which is folding of the secondary structure on itself into ball-like substance, so that the exterior is mainly of the hydrophilic amino acid and interior is hydrophobic amino acid. And this tertiary structure is required for activity of the protein. So protein will be active only in the tertiary structure. Then there is quaternary structure, in which there are many units of tertiary structure, same or different, which come together to form a functional protein. And tertiary and quaternary structure consist of same type of bonds like hydrogen bond, disulfide, hydrophobic bonds, ionic or electrostatic bonds, and van der Waals forces. So that was about structures of protein which you should know. Next is denaturation. So what happens? So as we have discussed, that this folding of protein is required for its functioning, so that it can become active and participate. But what will happen if they are denatured? Their folds are removed, so they will again become non-functional. So phenomenon of disorganization of native structure of protein such that it loses its biological activity is known as denaturation. So if the folds are removed, the denaturation will occur and the protein will lose its activity. So it will lead to unfolding, loss of secondary, tertiary and quaternary structure, but primary structure will remain intact. So this is important. In denaturation, secondary, tertiary and quaternary structures are lost, but primary structure remains as such. So in what conditions denaturation can occur? It can occur under physical conditions like heat, violent shaking, X-rays or UV radiation, or under chemicals like organic solvents, detergents, strong acids, metals and enzymes. So all these can lead to denaturation of proteins, which will lead to unfolding of proteins, so in such a way that their function is lost. But denaturation is also important as it helps in digestion. Denatured proteins are easily digested due to increased exposure of peptide bond to enzymes. So peptide bonds are directly acted upon by enzymes and they are split into amino acids, so digestion is easy. That's why cooking of food make proteins easily digestible. So that's why when we cook food, the proteins are denatured and we can easily digest them. Why we can digest them? Because there is unfolding of the protein and there is straight exposure of peptide bonds that are present in primary structure to various enzymes, and they can be digested easily. So now we have discussed that protein folding is very important. So unique structure of protein involves the folding of its polypeptide chain into characteristic 3D structure, and this is essential for biological function of protein. And normally the proteins fold by themselves because of the primary structure. The primary amino acids make bonds with each other to far away amino acids also to form a secondary, tertiary and quaternary structure and become folding. But sometimes in a body, the folding require assisted. So assisted folding is some proteins do not fold spontaneously. They require some accessory proteins which helps them to fold, like protein disulfide isomerase, prolyl cis trans isomerase and chaperones. So these are the proteins which helps the proteins to get folded so that they can become biologically active. So chaperones are also known as heat shock proteins. How do they work? They interact with the partially folded or improperly folded protein and facilitates the correct folding. So it helps in correct folding of the protein so that they can be functional. Like we know that the protein is folded in tertiary and quaternary structure to be active. So most of them have ATPase activity and need ATP dependent binding. So chaperones or heat shock proteins assist proteins to get folded so that they can become active. So there are two types of heat shock proteins which you should know. One is heat shock protein 70 and second is 60. 70 is found abundant in cells stressed by heat. So bound to hydrophobic region of nascent polypeptide and shield them from the solvent. So first one are heat shock protein 70 and second is 60, which has a pocket in which unfolded protein binds, and this covers it like a lid and provide appropriate environment for folding. So these two types of proteins, heat shock protein 70 and 60, helps the proteins to fold so that they can become biologically active. So what will happen if these proteins are not folded properly? So we have discussed that the folding is very important for their normal functioning. But if folding is not proper, what will happen? It can lead to various disorders like amyloid diseases as seen in Parkinson's or Alzheimer's disease. So this is due to misfolded beta amyloid peptide accumulations resulting in neurodegenerative disorders. So if proteins are not folded properly, they will not be active. Rather, they start accumulating and can result in neurodegenerative disorders as occur in the case of misfolded beta amyloid peptide. And it can lead to Parkinson's and Alzheimer's disease. Then other disorder is Prion's disease like Creutzfeldt-Jakob disease in humans. So here normal cellular prion protein is replaced by infectious protein and alpha helix are replaced by beta sheet. So folding is not in that way that is required, which will lead to this disorder. Then in hereditary emphysema there is correct but slow folding of the mutated alpha antitrypsin. As a result, unopposed action of elastase will act on lung elastin and will destroy the lung elastin, so resulting in emphysema. Then similarly in cystic fibrosis, there is mutated CFTR gene, a membrane chloride channel protein, which fail to dissociate from chaperones. So chaperones try to fold them, but they fail to dissociate. Thus, because they are not able to dissociate, they can't reach to their destination, resulting in their deficiency, or they can't act properly. So these are the various disorders which are disorders due to improper protein folding. Then students, one more thing which you should know is protein electrophoresis. So we have discussed that protein electrophoresis, we can separate the proteins based on their negative charge, size and negative charge. So normally when electrophoresis is done, it separates it into 5 bands: albumin, alpha 1, alpha 2, beta and gamma. So this is a normal pattern of electrophoresis of plasma proteins. But if there is any difference, any disorder, what will happen? We will get different electrophoretic pattern, and on the basis of that, we can make the diagnosis. Like in multiple myeloma, we will get M band between beta and gamma subunits. So this M band, if there is present in electrophoresis, it help us to make the diagnosis of multiple myeloma. Similarly, in chronic infections, there is increased production of immunoglobulin. So gamma band is increased in size. So we can make a diagnosis of chronic infection. Similarly, nephrotic band, there is hypoalbuminemia, there is prominent alpha 2 band. So, and albumin band is less prominent. So, in this way, with help of electrophoresis, we can make the diagnosis. Like in cirrhosis or liver, there is increased gamma globin. Nephrotic syndrome, increase in alpha 2 fraction. In multiple myeloma, as we have discussed, there is M band between beta and gamma. And in A gamma globulinemia, there is no gamma globulin. Chronic infection, there is broad base increase in gamma band. So on the basis of different electrophoretic patterns, we can make the diagnosis. Now the question is, a group of proteins that stabilize unfolded or partially folded proteins by exhibiting ATPase activity are, so we have already discussed, they are known as chaperones. So chaperones help in assisted folding of the proteins. Now the important points to remember from this lecture are, first of all students, you should know about the primary, secondary, tertiary and quaternary structure. So we have discussed that primary structure is linear joining of the amino acid with help of peptide bonds, and secondary structure is formation of hydrogen bond which helps to form either coils or beta pleated sheets. Then there are supersecondary structure which joins the primary secondary structure with help of loops or bends. And then there is tertiary structure, which is folding of the secondary structure into ball-like structure with the help of many bonds. And then quaternary structure, in which there are number of subunits of tertiary structure which come together to make a functional protein. So a slight change in primary structure will lead to modification of amino acid and can lead to disorder. So the sequence of amino acid is very important because it will dictate the protein folding also and the type of protein to be formed. And even there is change in one amino acid, it will disrupt the structure of the protein and can lead to loss of biological activity. Then denaturation we have discussed, that is, it is unfolding of the protein due to physical or chemical conditions, and but it do not affect primary structure. It affects secondary, tertiary, quaternary, but primary structure is not affected. That is, peptide bonds are not broken. So this is important, you should remember. Then supersecondary structure is also known as motifs or folds, are combination of secondary structure. So they are in between secondary and tertiary structure. That is, they combine alpha and beta plated sheets either with loops or bends, and they are intermediate. Then tertiary structure is essential for biological activity. So enzymes and myoglobin are in tertiary structure because they are required for their biological activity. Then chaperones, you should know, they interact with partially folded or improperly folded nascent proteins to correctly fold the proteins. Then you should know about electrophoresis, which separates 5 bands of different proteins in serum like albumin, alpha 1, alpha 2, beta and gamma. And if there is some abnormality in this electrophoretic pattern away from normal, it helps in to reach the diagnosis. Like in multiple myeloma, we get M band. So that is all about structure organization of protein. So we have covered in protein chemistry about classification of amino acid. Then we have discussed about ampholytes and isoelectric point, and this last lecture we have discussed about of the structure organization of proteins, how the primary, secondary, tertiary and quaternary structures are important. Then we have discussed about denaturation, how proteins are folded, and what are the disorders which can lead because of improper folding of proteins, and various electrophoretic patterns which helps in diagnosis. So that's all for this lecture. Keep studying. Thank you so much.